Hi, everybody. I'm going to finish up the cell lecture that we stopped last week, and this is going to be part of your first quiz, quiz one. So let's get started. This was going to be part of a worksheet, so I'm going to kind of go through it and talk about what we would have done in the worksheet, and so that you could maybe understand this better. So this is about cell transport. And the first thing we're going to talk about is how molecules, particles move around in space. And so I'm saying particles because it could be an ion, it could be a molecule, it could be an atom. And in this example, we're looking at this beaker over time. And so here you have time 1, 2, 3, and 4. And there is a beaker full of water with a sugar cube here at the beginning of the time. Now, if you're looking at this and thinking about sugar as the molecule that we're thinking about when we're talking about concentration, if you put a square around the sugar at its highest concentration, you would probably, oh, trying to get it to, there we go. You would have a square around this cube of sugar. That's where the sugar would be at the highest concentration. And in this beaker, the area that would have the lowest concentration of sugar, if we put that in a circle, would be around here. There's other parts in that beaker that would have a low concentration as well. And if we drew an arrow from the area from highest concentration of sugar to the area of lowest concentration, it would look something like this. This is called the concentration gradient. And the concentration gradient is going to be defined as an area that has a high concentration in one part of that area and a low concentration of that substance in another part of the area. Now, these molecules, these particles are moving around randomly. It's called Brownian motion. It happens all the time, especially in a liquid like this. And so as those molecules of sugar are moving around, they're more likely going to be moving in a net movement towards areas of low concentration, just because that high concentration has a lot of molecules there, so that they're more likely to go in the direction where there's not going to be much. There's going to be movement of molecules in the low concentration area, but they're just so few that the chances of a lot of them moving towards the high concentration area is pretty low. And as you can see, as we go along the time series, those molecules of sugar are going to get more and more dispersed, going from the high concentration and moving as a net movement into the low concentration area. And then here in um, time 4, you can see that they're all evenly dispersed. They're still moving around. There's just not a net movement, right? There's no concentration gradient here anymore because there's no area of high concentration and an area of low concentration anymore. That is what diffusion is. So think about diffusion and its definition and an example in the body. So diffusion is going to be the net movement of particles from areas of high concentration to areas of low concentration via this random movement. An example that this happens in the body would be the movement of gas and carbon dioxide going from areas of high concentration to low concentration. So here in this example, we have a capillary that's carrying blood. There are high concentrations of oxygen in the blood, low concentrations of carbon dioxide. So that when they get to tissues that have low concentrations of oxygen, there's going to be a net movement of oxygen into those tissues. And these tissues are going to be doing cellular respiration, which produces carbon dioxide. So there's going to be high concentrations of carbon dioxide. These are going to have a net movement into the bloodstream because those areas have low concentrations of carbon dioxide. Now we're going to think about facilitated diffusion. Now, it's still going to be a movement of materials from high concentration to low, but it's going to go through a channel. That's why it's called facilitated. This is going to be involving particles that aren't able to move through cell membranes without having a channel or a carrier of some sort made out of proteins in the cell membrane. So it's still a movement that due to this concentration gradient from high to low concentration. And there's no need for the cell to provide any energy for this motion to occur. So in this example here, we have these carrier proteins where glucose molecules can attach. And you can see here on the outside of the cell in the extracellular fluid, there's a higher concentration of glucose molecules. And here there's no glucose molecules, so here's a low concentration. So it's much more likely that glucose molecules are going to attach to the protein here, and then that will cause a change so that the carrier is going to switch and open up into the cell and bring that glucose in. So this can happen with certain ions because ions cannot pass through the phospholipid bilayer. They must go through protein channels and the movement of glucose. There's a special kind of diffusion that occurs with water when there is a semi-permeable membrane. Now, semi-permeable means that there's certain materials that are able to go through the membrane and other materials that are not able to go through the membrane. Water can go through the membrane because cell membranes are filled with little pores called aquaporins, which are specifically there to allow water to move freely back and forth through the cell membrane. There are lots of solutes, large solutes, that can't cross this membrane. And so when this happens, we have this process called osmosis. Now, this is an image from your book, and it's showing how in this part of the tube, there's a semi-permeable membrane, selectively permeable, which allows water molecules, here illustrated with the small blue dots, to go through the membrane. These larger solutes here are not able to go through this membrane. Now, if you look carefully, you can see that there's a lot of water molecules here. And on the other side, on the B side of the tube, there's not as many water molecules, and there's quite a few solutes. This means these solutes are dissolved in the solution because water molecules are surrounding these solutes. So they're not really moving freely because they're involved in surrounding these solutes. There's a lot more free moving, randomly moving water molecules on this side, on the A side. And so there's going to be a net movement of water into the B side, so much so that it's going to cause the water level to go up here in the B side of the tube because all this water has gone through here. Now it's at equilibrium. There's no net movement back and forth. And so they're going back and forth, but there's no more net movement going into the B side. And you can see, yeah, the amount of the concentration of water molecules is about the same on both sides, and this concentration of solutes is the same. If we were to try to get this back so that they were evenly at even levels again, if we put the force that would cause water molecules to rush through this selectively permeable membrane, that applied force would equal the osmotic pressure. That's why there's osmotic pressure up here. This is an important concept. This is an important process in our bodies. When we have blood that has a lot of water in it, also have large proteins inside the blood vessels, that means water is going to go into the blood vessels from the tissues. This is going to return back into the tissue from the tissues into the blood, and this allows for the blood volume to be maintained at a pretty good level. And you'll go into that more when you do the circulatory system. There are your notes. And another thing to think about when we're talking about concentrations of solutes in a solution is tonicity. So this is an image from your book showing different tonicities that these red blood cells are in. And so let's go through each one of these. First, we have isotonic. Iso means equal, like isosceles triangle. Remember that from geometry class? Iso means equal. Tonic is referring to the amount of solutes, the solute concentration. Specifically, it's the relationship between the solute concentration of the fluids that surround the cell. So here in this blue area, that solution, and the amount of solutes that are within the cell. So in this case, isotonic means that the concentration of solutes within the cell is the same as the concentration of solutes outside the cell. That means water is moving pretty equally back and forth in and out of the cell. There's no net movement of water. This is a happy cell. This is where cells are going to be functioning their best. So now let's move to hypotonic. Hypo means below. So something that's below the amount. Now, it's hard to see here, but see these little purple guys? Those are the solutes. So you can see that there's some solutes here inside the cell, but there are no solutes outside. So this is like a cell that's inside a beaker full of very pure water, right? Distilled water here. That means because there's less solutes outside in the solution that the cell is in, that means it has less solutes, means hypo, below, solutes for tonic. That means that there's a higher concentration of water outside the cell than there is inside. That also means then that there's going to be a net movement of water inside the cell. And here's what happens to cells that are in a hypotonic solution. They start to swell up. And they can swell up so much that they're going to burst. And so we call that hemolysis, in which hemo would be the blood cell and lysis means cutting. Finally, we get to hypertonic. So here we see a cell that has some solutes inside, but you can see that the material that's outside that solution has a lot more solutes. Hyper meaning more, tonic referring to the solutes in the solution. So there's more solutes in the solution. Think of this as really salty water. In this case, there's a higher concentration of water molecules inside the cell than outside. And so that's going to cause a net movement of water out of the cell, causing it to shrivel up as it's losing fluids. And now let's go to active transport. As you can think when you hear the word active, this is going to require energy. And so it's going to be used when a cell needs to move material from a low concentration into a high concentration. And this often will be done by a molecule called a pump. And think about pump needs energy. And so here with the sodium potassium pump, we can see in the extracellular fluid outside the cell, there's a low concentration of potassium ions. K plus here is for the potassium ion. And this pump is going to take the ions, the few potassium ions that are in the extracellular fluid, and pump them into the cell, which has already a high concentration of potassium ions. The other thing that happens in this pump is that the few sodium ions that are in the cell are going to be pumped out into the extracellular fluid, which you can see as a high concentration of these sodium ions, this Na. Oh, here we go, Na plus. This requires energy. So that energy transfer molecule, ATP, is being used and turning into ADP because one of the phosphates came off to energize this potassium pump to work. Examples for this would be certain sugars, amino acids, and ions that are needed to be at different concentrations inside the cell than they are outside the cell. And now we'll go to endocytosis and exocytosis. Endo means going in. Cytosis means a movement of a cell. Cyto means cell. Exocytosis, moving out. So going out of the cell. So there's some examples. In your book, it shows both processes here. So exocytosis would be when materials that are going into, that are in a vesicle are going to, that vesicle membrane is going to fuse with the membrane of the cell and release material into the extracellular fluid and to the outside of the cell. Endocytosis, materials that the cell needs are going to attach to proteins that are in the, that are on the cell membrane here. And then when there's enough of them, that cell membrane is going to pinch and form a vesicle. And then that material is now can be used inside the cell. So endocytosis, movement of large matter via vesicles into the cell, exocytosis, movement of large matter out of the cell. An example in the body would be white blood cells that are going to be uh, trying to protect the body from any kind of potential pathogens. They can form these pseudopodia that surround any kind of potential pathogens, and then they're going to eat them up. Lysosomes are going to come and fuse with the vesicle that has the pathogen, and then that pathogen is going to get digested. For exocytosis, an example would be neurons that are sending a signal from one neuron to the next. They need to send a chemical signal from this neuron here to the receiving neuron, the postsynaptic neuron. Little vesicles are going to be filled with neurotransmitters, proteins that are made by this cell, and those vesicles are going to fuse to the cell membrane, and then those um, neurotransmitters are going to diffuse from high concentration to low concentration to receptors here on this cell membrane. And that's how they're going to send the signal to the next neuron. Finally, in this chapter, they talk about the cell life cycle. I know it's a really long chapter. And the cell life cycle, basically, you'll have a cell that's happily in G0 doing its function, and then it might get a signal that it's time for it to divide. And so it needs to make more of these cells. The body needs more of these cells. If that happens, it's going to go into the G1 phase, where it's going to start preparing um, the cell for cell division. And then it's going to go into the S phase. S phase, so I think of G1 as the first growth phase, and then the S phase is the synthesis phase of DNA. So if the cell's going to divide, it needs to have duplicates of lots of things, including DNA. And that's what's going to happen in the S phase. And then we get the G2 phase, where a little bit more growth happens in preparation for cell division. Finally comes mitosis. Well, not finally, there's also cytokinesis. But mitosis is specifically the division of the duplicated DNA chromosomes. And then cytokinesis is the division of the rest of the cell, of the cytoplasm with all the organelles inside. So in mitosis, which is used for cell growth, for the growth of the organism, for replacement of cells that have been lost or cells that have been damaged. And so we'll have cell division where you have the original cells, the chromosomes of the nuclei are going to divide, and then the cytokinesis, all the organelles are going to divide, and then you're going to have two daughter cells. Mitosis is specifically the separation of the sister chromatids. So before the cell goes into S phase, each chromosome is going to be one strand, like this one strand. But during the S phase, another strand is made so that this strand is identical to this one in its genetic makeup. We call these sister chromatids. During mitosis, these chromosomes are going to go and line up in the middle of the cell here. And then you're going to have this metaphase plate. And then you're going to have those separate to two ends of the cell so that now the sister chromatids have separated. One copy of the nuclear of the DNA is going to go on that end, and then oh, my dog's barking, and then the other end is going to come this way, and then you're going to have the nuclear membrane forming again around the chromosomes, and Coco, hush up, and then you're going to have the cytokinesis happening here during telophase, and that's it. All right. I hope this was helpful. I hope I didn't talk too fast. I'll see you later. Bye-bye.